We're now going to move on to renal physiology. This is the key mechanism that helps to maintain a number of necessary homeostatic mechanisms so that the body's fluid and electrolyte and also pH levels are properly controlled. So it's a really key mechanism for overall function of the body. There will be 3 sections for, this is Unit 8, this is Unit 8A, and this video is Part 1 of Unit 8A, or Lecture 8A, I'm sorry, not Unit. Lecture 8A, Part 1. So for renal physiology, first we'll go through, as we have in the past, we'll go through the anatomy, and then I'll start talking about the physiological functions and the regulation of those. Renal physiology is complicated. There are a number of regulatory mechanisms that can operate, and it takes some level of understanding the overall picture. And please understand that when we talk about the physiology in the kidneys, we're always looking at fluid and electrolyte levels, so they fluctuate with time, and really you need to have a good feeling of the overall mechanisms that regulate it in order to understand it. And you need to understand what kind of changes will trigger those responses, what kind of changes in a person's physiology will trigger the regulatory mechanisms that affect fluid, electrolyte, and in Lecture 8C we'll talk about pH balance. Okay, now you should always keep in mind that cells are surrounded by extracellular or tissue fluid. You know, the cells have fluid inside the cytoplasm, that's the intracellular fluid. They're also surrounded by extracellular fluids, and there are 2 extracellular fluid compartments: the interstitial fluid, which is the tissue fluid between cells, and the intravascular fluid, which is the fluid in the part of the blood. The osmolarity of the fluids will determine the volume and osmolarity of the the osmolarity of the tissue fluids will determine the volume and osmolarity of the intracellular compartment. These different compartments, intracellular and the 2 extracellular, which are interstitial and intravascular, are all in contact with each other, either directly or indirectly, and will affect each other. There may be some time delay, but if the osmolarity of the blood plasma drops, then fluid or water will start moving into the interstitial environment, and with some delay into the intracellular environment. At the same time, of course, the body would respond and try and put out more fluid as urine, right? A larger urine volume. But nevertheless, they affect each other. Osmolarity is different from molarity because it looks at the number of moles, the quantity of molecules of solute, rather than or dissolved particles rather than just particles. So some compounds will dissociate in solution, so you have to take that into account. Ionic compounds, like different salts, can dissociate once they're into solution. So 1 mole of NaCl, once you put that NaCl or sodium chloride, once you put it into solution, it dissociates into its individual charged ions, sodium and chloride ions. So sodium is positively charged, chloride is negatively charged. They're in the solution. Therefore, there are, when you talk about osmolarity, that 1 mole dissolved into water will be 2 osmoles of solute particles in the fluid, in the solution. So 1 molar NaCl is a 2 osmolar solution, as pointed out here. So you have to keep that in mind in terms of osmolarity. You realize that they dissociate in solution. Non-ionic compounds don't dissociate. You'll get 1 osmole of the solute per 1 osmole of solute not in solution. Okay. Now, in terms of electrolytes, these are compounds that can dissociate into charged ions when dissolved in water. And cells require very precise concentrations of electrolytes internally and externally. So intracellular and extracellular fluid compartments have to have particular concentrations of electrolytes in order to function in a balanced manner, in order for them to function normally. And maintaining that electrolyte balance is, in fact, crucial to survival. And electrolytes, typically we're talking about salt balance. I don't think of salt as only NaCl. There are a number of different salts. It's not just referring to NaCl, although sodium, Na, the ion, is particularly important, and you'll see that soon enough. Salts do enter the body by ingestion, and they can be lost by perspiration or in the feces or as urine. And they include salts. Yes, NaCl is a key salt that we have in the body, but there are other salts, and it also includes acids and bases. Although we're going to talk about acids and bases separately, and we'll discuss that in lecture 8C. Okay, so if we look at fluid and electrolyte balance in terms of the sodium content, sodium holds a central position. Sodium salts account for 90 to 95% of all the solutes in the extracellular fluid compartment. So normal extracellular fluid solute concentration is 300 milliosmoles, and sodium contributes 280 of that. So we're talking about greater than 90%. It's actually, I guess, 93% of the total is attributable to sodium cations. I'm sorry. So 280 milliosmoles of the 300. It's the single most abundant cation in the extracellular fluid, and the only one that exerts significant osmotic pressure. In other words, it can pull water across semi-permeable membranes that make up all of our tissues, that are present in all of our tissues. Semi-permeable membranes are the cell membranes, right? That dominant role is the key idea, and why we say that sodium has a dominant role is, again, because it has significant and can exert significant osmotic pressure. And sodium ions are constantly being pumped out of the cell against their electrochemical gradient. So sodium concentration is much higher outside, and that's the chemical gradient of sodium, the actual concentration. And the inside of the cell, cells typically on the inside are negative. They have a negative membrane potential, and even though opposite charges will attract, the sodium which is positively charged, is being pushed out, pumped out. That's a energy requiring process. It requires ATP to operate the sodium-potassium pump. So sodium is higher outside. It does leak in slightly into the cells, but for the most part, the cells are impermeable. They are certain tissues, certain cells can become temporarily permeable to sodium if sodium channels open. So excitable tissues like muscle and nerves, they do have sodium channels which can open if given the right signal. Sodium concentration in the extracellular fluid remains normally very stable and is regulated by homeostatic mechanisms to be at a stable level. If there are any changes in the intravascular sodium level, the plasma sodium level, they can affect the actual plasma volume. They can affect blood pressure, because when you change the volume, you will affect the blood pressure in the intravascular space. If you have an increase in plasma volume, typically you get an increase in blood pressure, and vice versa. The plasma volume, of course, the plasma is the intravascular fluids, part of the extracellular fluids. The other type of extracellular fluid is interstitial fluid, and again, sodium has an effect on that. And then there's the intracellular fluid. Remember, there are 3 fluid compartments: intracellular and these 2 extracellular fluid compartments, plasma and interstitial fluid. So these are the extracellular compartments, and this one here, intracellular fluid, is the intracellular fluid compartment. And all 3 affect each other. They are affected by changes in sodium levels. If there's an increase in the sodium level in the plasma, that will pull water by its osmotic ability, osmotic pressure, osmotic potential. It'll pull water across from the interstitial fluid, and that will trigger some fluid movement out of the cells. And it can go the other way. If the sodium level drops in the plasma, for example, then fluid can move from the intravascular space into the interstitial, and from there into the intracellular. So any shift in sodium concentration actually in any of the compartments would cause the movement of water across from one compartment to the next. The other thing about sodium is that, and this is I think the only thing right now that we're going to talk about in relation to acids and bases, the other thing is that control mechanisms, renal mechanisms that control pH levels, are always coupled to sodium ion transport. In order to change pH, you have to move sodium ions also. I want to remind you again, reinforce this idea of semi-permeable membranes and diffusion versus osmosis. So we're talking about, in general, osmoregulation. Diffusion is the movement of a substance along the concentration gradient, from regions of high concentration to regions of low concentration. And diffusion, the solutes, if the membrane is semi-permeable, which you do have in all of your tissues, you have all of those membranes are semi-permeable membranes. If the molecule can diffuse across, then they'll move from areas of high concentration to lower concentration along their concentration gradient until the equilibrium is reestablished. Here you have 2 different solutes, and here you have 2 different solutes on either side of a semi-permeable membrane, and they will, as long as the membrane, the molecules can, if these molecules can diffuse across that semi-permeable membrane, then they will establish an equilibrium in concentrations as shown at the bottom here. This membrane is permeable to these particular examples of molecules, but not permeable necessarily to all different kinds of molecules, and therefore and that's why it's called a semi-permeable or selectively permeable membrane. Now that's diffusion, and that's diffusion of the solutes. Osmosis is quite different. Osmosis is actually the diffusion of the solvent, the water. again through a selectively permeable membrane. Some membranes are selectively permeable. In this case, if we're talking about something that cannot move, it is impermeable. The membrane is impermeable to the molecules that we're using in this example. And most of the time, for most cells, sodium is an example of that. Then water will move instead of the solute. The solvent moves, the water moves, and in this case, with osmosis, of course, it's water. And the water will move from its area of higher concentration. Here on this side, water is in higher concentration. Okay, so it moves because the solute can't move. These dissolved particles here, they can't, on this side, there's a higher concentration than on this side. So water will move in order to try and balance the two. So keep that in mind. It's either the solutes moving if they're diffusible across the membrane, or the water moving by osmosis if a solute is not diffusible. Okay, we have excretory organs, the kidneys, 2 kidneys. They produce what we excrete, which is urine, and they control the osmolarity of fluids in different compartments, not only the osmolarity but also the total volume, and in that way they also help control blood pressure. We excrete solutes that are in excess. For example, if you have more salty food, so there's a higher amount of NaCl in the food, you'll excrete the excess. Kidneys will do that. They don't do it instantly, they take time. But essentially, you have to keep in mind that the kidneys, what they're really doing is constantly changing as a reflection of the state of your body. If you take in more salt, you'll tend to put out more of it, and you'll tend to try to conserve water somewhat, or you'll get thirstier. So you're always constantly trying to adjust back to a 300 milliosmol level in terms of osmolarity of the fluids. They do, however, always try to keep or conserve, not excrete any solutes that are important, like amino acids and glucose. These normally, very little of them get into the system, into the tubular system in the kidneys that'll lead to the bladder. Well, I'll talk about this later. If any do get across into that tubular system leading towards the bladder, then they will be normally always absorbed, so they do not appear in the urine. You tend to conserve these kind of solutes. The other thing that the kidneys do excrete is excess nitrogen, and we do have quite a bit of excess nitrogen, and the major source of that is from protein. Amino acids do have, every amino acid in protein has an amine group associated with it, and we take in far more than we need and have to excrete it. Okay, in terms of those kidneys or excretory organs, salt, water, and nitrogen excretion mechanisms are operating in order to keep a proper balance. You can hold in salt or let more salt out, hold in more water or let more water out. Nitrogen is a one-way trip pretty much. It's in excess, and in the diet, the intake is in excess, and therefore it's strictly excretory in nature. But the mechanisms involved in these maintenance of salt, water, nitrogen appropriate levels depend upon the environment that the organism is living in, whether it's a terrestrial organism like humans, or if it's aquatic organism in freshwater or saltwater, the mechanisms are in fact different. In humans, excretion involves 3 processes. These 3 processes are what the kidneys do. they carry out filtration, resorption, and secretion. Filtration of the blood plasma is carried out in the kidneys. It is really dependent on appropriate blood pressure within the capillary structures, these tufts or round balls of capillaries that are found throughout the kidney, and I'll show that to you in future diagrams. They're called glomeruli. That's plural for glomerulus. These are capillaries, small round tufts of capillaries that function as the filtration membrane, and fluids with their solutes get filtered across into the tubular system of the kidneys that eventually lead out of the body. So the glomerulus capillary wall is the filtrar membrane, and that's where filtration is occurring in the glomeruli of the kidney. Fluid enters into a separate tubular system, and then that fluid that goes from the blood plasma gets filtered across is called the filtrate. And the composition of the filtrate is changed after that. It's altered significantly. Filtrate first forms and then is altered by two processes, resorption and secretion. So you get resorption of some molecules, secretion of other molecules. Some don't get resorbed, some just get excreted, some do get resorbed. You'll see. We'll talk about that in future slides. But those three processes are key for the function of the kidney: filtration, resorption, and secretion. All right, let's talk about the excretion of nitrogen, which is, after all, one of the major functions of the kidneys. We take nitrogen, excess nitrogen, in our diet. Our diet, of course, is made of the same kind of molecules that we are made of: proteins, carbohydrates, lipids, and nucleic acids. And it's the proteins and nucleic acids that contribute to that excess nitrogenous waste. Mostly protein. Most of the source of excess nitrogen is protein. As I said before, it's the amino groups, the amino group of each of the amino acids that's the waste or in excess. Initially, it's produced as ammonia. However, ammonia is highly toxic, and then it's converted, at least in mammals, it is converted to urea. Okay. Now, different animals, depending on their, as I said earlier, depending upon their the environment they live in, produce nitrogenous waste in different forms. Aquatic animals typically don't convert the ammonia. It simply diffuses away from the blood plasma across the gill membranes. So, and that's the example with invertebrates that live in water, and also the bony fishes. So these organisms that produce ammonia and release it as their excreted nitrogen are called ammonotelic organisms. Terrestrial animals, they have to detoxify the nitrogenous waste by converting it to something that's less toxic than ammonia, and some do it by converting it to urea, and others convert it into uric acid. Now, the nice thing about urea is it's a highly water-soluble molecule. So mammals, amphibians, and cartilaginous fish, those are the sharks and rays, all produce urea and excrete it in a soluble form. These are called ureotelic organisms. There is, however, certain groups of organisms or species that produce instead uric acid. Now, uric acid is highly insoluble. It does not go easily into solution at all in water. And birds, reptiles, insects, and a few cartilaginous types of fish produce uric acid. Now they can't pee it out as a fluid because it's not, so they put it out as a solid, and that's the white stuff in your pet iguana or your pet lizard's poo. Okay, so and also in bird excrement, that's the white stuff. Here's an example of a car covered in uric acid, which is highly corrosive, by the way. Now, something you have to keep in, and I wouldn't worry too much about here, this chemistry, but other than knowing that there are ammoniteric, ureotelic, and uricotelic organisms, which get rid of their nitrogenous waste as ammonia or urea. I don't worry about the chemical structure, or uric acid, which is quite insoluble. Humans are ureotelic, but they can and do excrete some ammonia and some uric acid. And some individuals produce more uric acid than others. And because uric acid is highly insoluble, sometimes they get deposits of uric acid in the joints, particularly in the joint of at the base of the big toe and the foot. And that causes a condition called gout, which is actually a type of inflammatory disease of the joints, and therefore I'm trying to find my, there it is, which is an inflammatory disease of the joint, and therefore is a form of arthritis. Gout, deposition of uric acid crystals in the joint. Now, uric acid can be put out in the urine, and when they take a urine sample in a tube, they'll spin it down, they'll centrifuge it, and they will look for uric acid crystals, whether they're present or not. Okay, let's describe some of the anatomy of the excretory system in human. There are, in humans, 2 kidneys. These are found in a position in the body called, they are retroperitoneal. They are not in the peritoneal cavity, they are behind it. Now remember, the peritoneal cavity, like other cavities in the body, is surrounded by membrane. There is a parietal membrane around the body cavity, and around the organs in the peritoneal cavity, there's a visceral membrane. So you have a parietal peritoneum and a visceral peritoneum. So the wall of the peritoneal cavity is covered by this membrane. The I'm sorry, just a second. The parietal peritoneum, and behind that, underneath that, more towards the dorsal side, are the 2 kidneys. They do produce urine and release it, they make a filtrate, release it into a ductular system where it undergoes resorption and secretion, and then it comes out of the kidneys as urine and goes into the ureter, and then down to each of the ureters from the 2 kidneys lead down to a bladder that stores it, and then the final excretion is through the urethra. So in mammals, the nephron in the kidney is the functional unit. It's the nephron that does pretty much everything in terms of making the excretion. And in each kidney you have approximately 1 million nephrons. So the total with 2 kidneys is 2 million nephrons. There are 3 major structures in a nephron. There is the glomerulus. This should be singular, not plural. In each nephron there's glomerulus. There are the renal tubules, and there are peritubular capillaries. It's at the level of the glomerulus, which is a capillary bed, a round knotted capillary bed, where the filtration occurs and the filtrate is formed, and then that filtrate fluid goes into the renal tubules and it gets processed. Either there's resorption of parts of it or secretion into it from the surrounding tissue. And there are peritubular capillaries that surround those tubules. And here you see a diagram showing the position of the kidneys and the retroperitoneal to the peritoneal cavity. The outer part of the kidney is all of this here. This area here is called the cortex. And the inner part of the kidney, all that is called the, let's do it in another color. All this area here, inside here, is called the medulla. All right. The glomeruli are positioned, the nephrons rather, are present. This is a vast exaggeration of a size of a, this is trying to show one, I'll show it later. Maybe in a later diagram I'll describe that. Hang on. It's hard to, okay. So the outer portion of the kidney is the cortex, the inner portion is the medulla. There are these pyramid shaped in 2 dimensions. They look like a triangle, but they're actually pyramidal in shape. They're the renal pyramids. Renal artery supplies blood into the kidney and the renal vein drains it out. Okay, so the outer cortex, the inner medulla, you have renal pyramids. These are positioned within the medulla in this way, and at the very tip of each pyramid, that's where all of the tubules are emptying. The tubular system empties right into the very bottom tip. Let me point it out to you. The bottom tip of each, right in. It's a little tricky changing colors sometimes. The bottom tip is where the urine of each of these pyramids is where the urine goes into the collecting system that leads to the ureter that heads down to the bladder. So as I said, there are 10 million nephrons in each kidney, and the glomeruli, that capillary bed where the filtration occurs, is all of those are in the cortex. So if we look at a magnification, and I have to point out to you that there are 1 million nephrons in each kidney, and so just for illustrative purposes, this artist has made a nephron much, much, much bigger than it actually is. So they're much tinier. But there's the nephron. The nephron is made up of this glomerulus here. Let me get another color if that'll, no, that doesn't work. So here, okay, that's the glomerulus inside the glomerular capsule, also called the Bowman's capsule or glomerular capsule. So the capsule is the beginning of a tubular system. The filtrate, which is the filtered product from the plasma fluid that gets filtered across the glomerulus, the capillary bed of the glomerulus, goes into that tubular system and then goes through into the capsule, the beginning of the tubular system, and then goes through this winding tubular structure, okay, in this kind of a way, comes back up and then goes down into the collecting duct, down to the tip of the pyramid, and then out. This initial part of the tubular system, of course, is the Bowman's capsule, and right after that, and that's always present in the cortex. After that, the first tubular section is called the proximal convoluted tubule, and then diving down into the medulla, you have the loop of Henle, and there's a descending arm of the loop of Henle and an ascending arm back up towards the cortex. The loop of Henle is surrounded by peritubular capillaries that have a particular name. It's the vasa recta. And they lead back, the loop of Henle leads back to the distal convoluted tubule, which always comes back near to the glomerulus and glomerular capsule of the nephron they originate from. And then the distal convoluted tubule empties into a collecting duct. Several different, and that's shown by these truncated parts here. Several different nephrons will empty into each collecting duct. There's many collecting ducts, all running down through this renal pyramid. Okay, it's full of collecting ducts, all leading to here, the very tip where it empties out its final product, the urine. Okay. And here you see a prepared specimen. It's a, I think this is a scanning electron micrograph, not a light light microscope micrograph. It's an electron micrograph. But what you see here are many glomeruli. Each of these round tufts is a capillary bed of the glomerulus. Each one is a glomerulus, many glomeruli. What they've done here to prepare this specimen is that they injected latex into the vascular system. They injected latex probably into the artery, and the liquid latex fills up all of the arteries and capillary beds, arterioles and capillary beds, and then solidifies, and then they digest away all other tissues. So everything else has been removed, and the latex, of course, solidifies, and they digest away all the other tissues. So you're left with this skeleton cast, essentially, of the vascular system of the kidney, the nephrons here, these glomeruli rather. Okay, so as I said, the first part of the tubular system is the glomerular capsule, and then it leads to that's not a very good color, is it? The glomerular capsule here, and leading then to the proximal convoluted tubule, and then the loop of Henle, and then the distal convoluted tubule, and then the collecting duct. And here you see a single glomerulus inside a glomerular capsule or Bowman's capsule. And remember, the glomerular capsule or Bowman's capsule is the beginning of the tubular system, and there you see the very, very beginning of the proximal convoluted tubule. So this glomerulus is filtering the plasma, and the filtrate is forming in this space here in the Bowman's capsule, and then heading down the heads down the proximal convoluted tubule. And the end result is that urine, final product urine, comes out here. Now, as I said before, in terms of blood vessels, it's a renal artery feeding into each kidney, and then the arterioles, it goes into smaller arteries and then arterioles, and they branch and they go into the cortex, and the arteriole leading to each glomerulus is called a afferent arteriole. The afferent arteriole leads in and then twists around to form the glomerulus inside the glomerular capsule or Bowman's capsule. And then the arteriole leading out of the glomerulus is away from the glomerulus is called the efferent arteriole. And then that goes on to form the peritubular capillaries and the vasa recta all around the loop of Henle and the tubular system. Peritubular capillaries. Anything that gets after filtration, that fluid goes into the tubular system, and all the processes involved, the two mechanisms involved in changing the character of the filtrate, that is reabsorption or secretion. Whatever gets reabsorbed or secreted is either going to or getting absorbed into or getting excreted, coming out of the peritubular capillaries and moving into the loop, or for resorption coming out of the loop of Henle or the tubules rather, and going back into the circulation. So the peritubular capillaries surround most of the tubules in the cortex, and then they dive down and they form the vasa recta around the loop of Henle. And so here in this organ, in this structure, when we look at the nephron, the simplest functional unit that carries out the major physiological role of the kidney, which is to form urine, perform fluid and electrolyte balance. When we look at the nephron, each nephron has two capillary beds. One is the glomerulus, and the other one is the peritubular capillaries or capillaries. And here you see another diagram showing the glomerulus here, fed by an afferent, now it's down at the bottom, an afferent arteriole here. Whoops. An afferent arteriole right there. Then the glomerulus, and then an efferent arteriole that then twists around and surrounds, not shown here, but surrounds all of the capillaries and the tubules and loop of Henle. So I think I'll stop here. This will be part 1 of lecture 8A, and then I'll go to wait a minute, I'll do 2 more slides and then I'll stop. This is a diagram showing 2 nephrons within the kidney, magnification of 2 nephrons, and it shows the characteristics that I just mentioned before. The glomerulus, there's another glomerulus, the Bowman's capsule around that glomerulus, then the proximal convoluted tubule all around here, back and forth. All this is proximal convoluted tubule, and then it gets into the loop of Henle, down and up. Some glomeruli have a much longer loop of Henle. They're more involved in making more concentrated urine than these. There are actually 2 major groups of nephrons: the cortical nephrons and the juxtamedullary nephrons. The difference is the juxtamedullary nephrons are near the junction. The glomerulus is closer to the junction between the cortex and the medulla. So it's a juxtamedullary, which means close to the medulla. And it also has a much longer loop of Henle that dips down into the medulla, much deeper into the medulla. Juxtamedullary nephron. Okay. Now, if we take a closer look, and this is the last slide I'm going to do on the anatomy here. If we take a closer look at the capsule with the glomerulus in it, and then in this artist's representation of the glomerulus, what he's done is shown the capillaries in cross section. So these are all connected together as one glomerulus, but there are capillaries that are shown in cross-section, and you can see the red blood cell, and they're exaggerated in size, but you can see the red cell inside. Remember, with capillaries, red cells essentially are going through, they're so small the red cells are going through in single file pretty well. And you see the space of the glomerular capsule, right? There's the glomerular capsule space, and there is a afferent arteriole bringing in blood to the glomerulus, and there's the efferent arteriole as a exit or draining blood out. So afferent arteriole and efferent arteriole. It's quite an unusual capillary bed because it's quite a high, much higher than normal pressure capillary bed. That's what allows for it to mediate filtration or form a filtrate. The fluids and solutes can get pushed out by that higher pressure. And then it leads to an arteriole as opposed to a venule. That's another difference. Now, I told you before that in the tubular system, there's the proximal tubule, there's the proximal convoluted tubule, the loop of Henle, and then the distal convoluted tubule. And I told you before that the distal convoluted tubule always comes back near to the glomerular capsule of the nephron that it originated from. And here you see a distal convoluted tubule shown in cross section. So it's shown as a round structure. That's in cross section. And there are specialized cells in the wall right here. You can see them here in this area here. These are macula densa cells in that distal convoluted tubule. The other specialized cells are the juxtaglomerular cells that are found around the afferent arteriole just as it enters into to form the glomerulus. So you have juxtaglomerular cells and you have macula densa cells. And together these form the juxtaglomerular apparatus, which I'll talk about later in terms of the physiological function of the nephron. All right, I think we'll stop there for lecture 8A, part 1.